Do you see my screen? Oh, no,
Yes.
it's gone again. Good. Okay. Then, first of all, I would like to say thank you for giving me the opportunity to present something, even though I unfortunately didn't make the trip to Hungary yet. But for the people that will also be there for the conference next week, you can see me then in person. And for everybody else, please email me or ask Harsh if you have further questions about what I'm going to present. So I'm going to continue talking about NV centers in diamond and sensing. And I've decided for today to show you one of our newest measurements that we did. And this is the first in vivo measurements that we have done with this technology. And I'm going to show you more specifically work on a C. elegans model for Huntington's disease. So what is the challenge? So Harsh already introduced a lot about radicals. Why are we interested in those? So they play a key role in a lot of different processes in the cells. So for example, cells use radicals to communicate with each other. If our immune system kills a bacterium or a virus, often radical generation plays a role. Radical generation also is important in a lot of different diseases, including cancer, cardiovascular diseases, infections. So it's very broadly applicable. But on the other hand, free radicals are short-lived and reactive, which makes it typically something that is difficult to measure and not used as much in the clinic as we would want to. So what can you currently do to measure radicals? So on the one hand, we have methods that basically measure not the radicals themselves, but how the cells react to radicals. Or we have a group of methods that basically measures the damage that they leave behind. And then we have the group that is closest to measuring direct, and this is fluorescent dyes. They react with the radicals and then change color, for instance. But a problem with this method, so first of all, these molecules bleach, so you cannot do long-term studies. They react with the radicals, so you basically influence the equilibrium that you want to measure, so you might get artifacts from that. And then the other point is these are small molecules, and typically you add the molecule, then you wait for something like half an hour, something like that, and then you read out. But the problem is in that time, a small molecule can diffuse very far and can travel basically anywhere in your sample. So you're losing the spatial information mostly. And what we do with NV centers is also optical, so we have a similar kind of signal, but then it is more local. And it measures the current status rather than the history of the sample. So what do we do? As Harsh mentioned already, we are working with defects that change their optical properties based on the magnetic surrounding. So what does that mean? We have basically something that is kind of like in an MRI, but measured in an optical way. So the particles essentially translate very small magnetic signal into relatively big optical signals that we can measure in a microscope. So now we have these small nanodiamonds that we put in the cells. By the way, to come back to the question, do we have plans to work with single NV centers? This is kind of difficult because we typically need the big ensembles to get the reproducibility and to actually see them within this high background environment. So that the short answer would be no. But now what we did here with the Huntington disease model. So first, a few words on Huntington disease. So this is a neurodegenerative disease, and it's characterized by formation of proteins that form aggregates. So it's called polyQ. And it's similar to what's known as the mad cow disease, where you have these prion-like proteins that form these aggregates and then become toxic. And what is some question in that regard is, okay, what is the role of radical generation in that process? And here we looked at this in vivo model. So this is C. elegans. This is a flatworm, and it's often used as a model because it's relatively transparent. We can also work with tissues. We did that before, but in this case, I'm going to show you the work in the transparent tissue. So how does it look like under the microscope? So these are the worms. In this case, the way we get the nanodiamonds into the worms is that we add the worms to bacteria. The nanodiamonds stick to the bacteria, and then the bacteria are food for the worms. So they eat the bacteria, so then the diamonds are in the gut, and then they can get out. And now this is how it looks like under the microscope. This is a glass bottom petri dish, and you can see here these worms. They are a few millimeters in size, so you can barely see them with your naked eye. And then on the bottom right, this is an incubator, so we can where it goes in, so we can keep during the measurements temperature and CO2 concentration and things like that constant in the medium. Now, what did we do with these worms? So first of all, we used two different kinds of worms. So we have a mutant where we have just YFP, so this is yellow fluorescent proteins, and Q0. So this Q0 means it doesn't have the protein aggregate, so it doesn't actually form the disease phenotype. And then we have the Q40 mutant, where we have yellow fluorescent proteins attached to Q40. So this is the disease phenotype, where we have these protein aggregates. Now, what we did first was we looked at our different phenotypes at the Q0 and the Q40 to check whether our disease phenotype is actually the phenotype that we want. So we checked, for example, are the sick worms really sick? And if you do that and you compare below with the Q0 versus the Q40, both of them, then you can see, for example, that the mobility reduces. Or you can also look at these worms when they swim, they have a characteristic bending, and you can quantify how much they bend. And then if they have reduced bending, they are not doing quite as well. And then you can also look at the metabolic rate. So we did measure the ATP level. That's also similar to what Harsh showed beforehand. So we can see how well these worms are doing. And this was basically done to confirm that the sick worms are actually sick and that we actually successfully made the Huntington disease model. Now, this is some imaging where we overlaid confocal microscope with bright field, and in red this is where you see the diamonds, and basically the structure here that is red, this is the gut. So you see that they behaved well and ate all these diamond-coated bacteria, so the whole gut is full of diamonds. Obviously, you don't want to really do sensing with these big aggregations here. So what we actually used for sensing is these particles here that are marked with the arrow that are a bit further away from the gut. So we don't measure this, but we measure in the cells in the intestine. And we also know that at the edge of the worms, this is where the muscles are. And we were interested in these muscle cells because, first of all, this is often where you see this aggregate formation. And second, these muscle cells are characteristic for early stage Alzheimer early stage Huntington generation. So we have these two locations. So we have the same worms, and then we look at the different location within the worm. So here is some data. So this is how the worm looks like under our microscope. So basically, we see all the parts that are in red. So very well visible is the gut. So this is the red line in the middle. And then you see some diamonds that are with some distance to the gut. And since you can see the worm itself, you can also tell where within the worm the sensing particles are. And then we did some measurement. This is shown here in B, where we measure, so N2, this is the wild type worm. So these are our healthy worms. And then SOD, this is a positive control. So here we induce some stress. And indeed, if you induce some stress, then you can see the stress response. And by the way, this is all data in the muscle here. And then you can look at the Q0. This is a control that is where you also have the YFP, the yellow fluorescent protein, but no protein aggregates. And the Q40, this is the experimental group. This is where we have the yellow fluorescent protein to visualize the aggregate and the protein aggregate to see what effect they have. And now you can do things like what we did here on the right, that you compare in the different groups. So here it's in Q0. What is the difference in the muscle and in the intestine, for instance? So this is the same worms, and we can compare what is the difference in one location and the other. Now here is the other location. So this is the same worm, but now the data is in the intestine. In the intestine, we can do the same measurement. So again, we did the same exposure of the worms. And what's really nice here is that we can really go to within the same worm before and after an intervention, can do a measurement. So this gives you a very powerful way to reduce the biological variability, because everything can be its own control. And this also gives you a nice way to reduce the variability from one particle to another, because you can do all the measurements before and after your intervention with the same particles. So here is the data for the Q40. So this is the protein aggregate. And here you can, again, compare between the muscle and the intestine. What we show here is semi-quantitative. So what we can do if we want to quantify this is you can calibrate it. But one thing about the calibration, we usually call it more an estimation, because it's very difficult to produce accurate concentrations of radicals in a test tube. So typically, we call it more an estimation, but then you can estimate what concentrations this corresponds to. And typically, it's something in the nanomolar range. So to conclude what I've shown you, so we can measure stress responses in living cells and with subcellular resolution. So in cells, this we now do quite routinely. We can do before and after measurement. So with this, you can disentangle what comes from my intervention and what comes from the biological variability or particle variability. It's organelle specific and it's real time. We have also shown this first measurement in living organisms. This is, of course, very nice in a transparent worm. In a tissue that is not transparent, we can also do that, but it's much more complicated. And there it's very difficult to have a context. In the worm, we can very nicely see where is the muscle, where is the intestine. But with a lot of like human tissue, for instance, it's more difficult to know the context of where you are measuring. Yeah, and in principle, you can also look at other applications where free radical generation plays a role. So if you are interested in this, we also have a company, it's called QT Sense, and we are selling this machine. So if you want one, you can let me know. And we're also hiring at the moment. So if you want to work with us, let me know. And then finally, this is my thank you slide. So I would like to thank a lot of people that funded this work. And of course, my research group, where you have Harsh as local representative there. So you can ask him further questions if you like. And